Welcome to part 2 of chapter 6. Again, the tour of the cell. This section we're going to talk about some major organelles involving what we described in the previous lecture as the endomembrane system, which involves a nucleus, endoplasmic reticulum, vesicles, Golgi, and so forth, as well as another heavily membranous organelle, the mitochondrion. Okay, I haven't mentioned this before, but in your book, they arrange these things by concept. So if you want to refer to the concept 6.4, there's a dedicated section in the book. You can kind of follow along that way, linking lectures with these concepts. Hopefully that'll help you as we work through this find material faster. So in this section, we're talking about the endomembrane system, which is the set of membranous organelles starting with the nucleus, running all the way out to the plasma membrane. A whole discipline of cell biology is devoted to understanding this dynamic because it's not a one-way thing. It is not merely this direction from the nucleus to the exterior. It's actually a trafficking situation. You can have things come out to here, and then it may move around different parts of, like, say, the endoplasmic reticulum, or to the Golgi and back. There's even a periodical journal called Traffic about this interplay. Sometimes we're bringing things into the cell in order to acquire nutrients or phagocytose cell debris in some cases, or we're secreting in other ones. So it's a complex interaction. It involves all of these structures: nuclear envelope, endoplasmic reticulum. And by the way, reticulum just is a patterning. That's why it's called that, is because when you look at it in a slide, it has a reticulated pattern. If you look at the skin of a giraffe, that's a reticulated pattern. There's also a reticulated python, but I didn't want to use a picture of a snake. But I think a giraffe skin is a good example of a reticulated pattern. The Golgi apparatus, lysosomes, vacuoles, and the plasma membrane all comprise this endomembrane system. The connection between these endomembranes is vesicles. And that's where the trafficking comes along, is that you're using motor proteins in some instances. The membranes have special targeting sequences and so forth, allowing for very specific and regulated interaction between these compartments. And here's the picture from your book. Again, the different parts, not all the parts are labeled here, but we certainly have the nucleus and we have the ER, and you have rough ER, which is primarily involved in protein synthesis, and then you have smooth ER, which is more along the lines of metabolism, protein modification in some cases. The vesicles connecting the ER with the Golgi, further processing and trafficking from the Golgi onto either the plasma membrane or other intracellular targets. So the endoplasmic reticulum, again, it's named that because it's primarily composed of membrane. So it's interior membrane, if you will, and it looks reticulated, especially when you look at it under a microscope. It's a huge membranous structure in cells. More than half of all the membrane in most cells is comprised of this, and membranes are very, very important for regulated interaction, and so makes it for a very, very important organelle. It is continuous with the nuclear envelope, though the membrane is different in its composition. So there are extensions off of the nuclear membrane, but again, these membranes are going to have a slightly different composition than these membranes, and so forth and so forth. Membrane composition is a hallmark of the different compartments. So two distinct regions. We have that rough ER, which is often involved in protein synthesis and embedding proteins in membranes for targeting to the plasma membrane in some cases, or are secretory proteins. You secrete them entirely into the interior of the membrane, and that begins the process of packaging them into vesicles. Smooth ER lacks those ribosomes. It's much more biochemical and storage in nature, and I'll explain what I mean here in a second. So here's the picture from your book showing the contiguousness of the nuclear membrane with the rough ER and then eventually smooth ER. And here are smooth ER examples and rough ER examples respectively. And like I said, they call it reticulum. Look on the left side, you can kind of see a vague similarity to the reticulated pattern that we observe, like say in giraffe skin. If you were to characterize the smooth ER in a couple of words, you would say metabolism and storage. Metabolism and storage. We synthesize lipids in the smooth ER. We metabolize certain carbohydrates in the ER, particularly disaccharides and small short-chain saccharides that need to be reduced to simple sugars. We can do certain types of oxidation and reduction reactions which are associated with what we would call detoxification. So if there are reactive molecules that are generated by the cell or are accidentally brought into the cell for whatever reason, the ER will have enzymes that are tasked with oxidizing and reducing those things, reducing the chemical form of reducing, so that these drugs are no longer toxic to the cell. However, that can also bite you. There are some precursor drugs that when they get reacted on in the ER can actually become toxic. So some interesting stories there. But another major function is calcium storage. So at the appropriate time, with the appropriate signal, the ER will release calcium into the cytosol and then in milliseconds pull it back in. It's a very, very short burst, and calcium is a frequent second messenger in cell signaling. It will activate proteins for a short period of time, and then you'll pull that calcium back in. Your skeletal muscle, a specialized form of ER called the sarcoplasmic reticulum, is tasked with releasing and reuptaking calcium ions. In talking about the rough endoplasmic reticulum, there are two words to keep in mind when it comes to function. Protein synthesis, that's because they have a membrane with these non-membranous inclusions, the ribosomes. So protein synthesis and protein targeting. Targeting means getting it to where it needs to be in or outside of the cell. So the rough ER has those bound ribosome inclusions, and it will, and secrete is a little bit premature here. It's a little bit premature, but in any case, if you see the word glyco associated with, like, for instance, proteins when it's covalently bound that way, or you see glycolipids or almost glyco anything except glycogen, I guess. In any case, if you see a word like this, other than glycogen, you can presume that it's either going out of the cell entirely, like it's being secreted, or it's being targeted to the cell exterior. It may still be embedded in the membrane, but the glyco part of it is going to be turned outward into an outward facing. It's not going to be cytosolic facing, if you know what I mean. Cytosolic facing. So let's look at this real quick. And so here's a rough ER, here's the membrane, and here's a ribosome. Proteins that are synthesized by the rough ER are either going to be targeted for non-cytosol compartments, that is to say other compartments inside vesicles, okay, or they're going to be secreted all the way out of the cell. Or also proteins that are embedded in the membrane, partially or completely, like protein channels or protein carriers, that sort of a thing. Free ribosomes, on the other hand, if they're free, where they're not associated with a membrane, most of those proteins being synthesized, and this is a relative minority actually, these proteins are going to be targeted for the cytosol proper. So the targeting part oftentimes involves that membrane. So depending on how embedded in the membrane, or whether it's going to be completely secreted or completely compartmentalized. And that's where the distribution, the targeting comes in. We're going to, if it's a secreted protein, it'll be in here, it'll be modified further, sent to the Golgi, probably modified a little bit more, we'll talk about that in a second, and then sent out of the cell. So it is a factory. The endoplasmic reticulum is a modification factory for targeted proteins. And closely related to the rough ER is the Golgi apparatus. This may be a bit naive to say this, but it's a good way to look at it. We have a distribution center for packages. So packages come in, they have labels on them, and those packages are then sorted according to what their label is. And that's a lot of what the Golgi will do. It's made of sacs and it has a receiving end, so vesicles come in on what we call the cis face, and they call it cis because it's closer to the nucleus. Okay, and then you have processing and targeting going on, sorting and as it were, and then you have a release side where new vesicles are emerging, and that's called the trans face. And so what we see is that you see further modification going on. Usually we're talking about things that have been added to the protein, or occasionally you're cleaving a protein, processing it for final pre-secretion, if you will. For instance, protein hormones sometimes get cleaved or modified in the Golgi and then stored in vesicles prior to release. So we're going to modify what's coming out of the rough endoplasmic reticulum in particular. Manufacture some things. I don't need to get into exact stuff here, but its major function is sorting and packaging into vesicles, either for transport around the cell or out into the extracellular space. And so the receiving side, cis, the shipping side, the trans, and you can kind of see the slight differences here. These vesicles are emerging off of here, and that's why it's much more reticulated in this area, because what happens is as the vesicles merge, they kind of form new pancakes, if you will. They often refer to Golgi as a sort of stack of pancakes. Our next organelle is known as a lysosome. And it is actually, in many ways, very analogous to those transport vesicles that we saw in the Golgi. But a lysosome, again, a very colloquial description here, is like the stomach of a cell. If your stomach is a walled organ, and inside that stomach you have a distinctive pH, it's very, very low pH in your stomach, and what you're doing is you're emulsifying and initiating digestion of material. A lysosome is very analogous to that. The pH in a lysosome is much lower than the regular cellular pH. It's full of enzymes, and its main role is to digest material into smaller biomolecules, break down polymers like carbohydrates or proteins or lipids, you name it. There are enzymes that are designed to create smaller molecules out of those larger ones. So they can, like I said, hydrolyze proteins, fats, polysaccharides, and nucleic acids. It's much more comprehensive as a cellular stomach than our actual stomach is. Our stomach is as much a biochemical barrier, and it initiates digestion as much as it does do the overall breakdown. We see more breakdown in the intestine. But in the case of the lysosome, that's all a cell has. And so it's going to do almost all of that process within the lysosome before those molecules are released or transported elsewhere in the cell. And so those enzymes work best in that acidic environment, just like you have enzymes that work best in different pHs in your digestive tract. One common process in cells, particularly cells like white blood cells, like neutrophils and other types of immune cells in the body, or glial cells in the brain, these cells are capable of something called phagocytosis, and this is very common in independent bacteria or independent cellular organisms like in pond water and so forth. So what happens is the nutrient molecule or the entity to be ingested binds certain sensor proteins on the surface of the cell. We call those receptors. And that initiates what's called invagination. It's where the plasma membrane begins to form and surround that entity with an entire plasma membrane, and it creates basically it creates a vesicle from the exterior that we will often call a phagosome. Okay, for lack of a better way to put it, we've brought in the plasma membrane and we've encapsulated that, and so inside this is still exterior environment relative to the cytosol. Again, a way of regulated interaction. What will happen is, as that phagosome is brought in, it will merge with lysosomes. And those lysosomes will then release, merge with that phagosome to create a phagolysosome. pH drops in here, those enzymes kick in, start breaking down lipids and polysaccharides, doing all this stuff. And what will happen is if the cell wants to retain those nutrients, it will. Otherwise, nutrients that it can't retain or won't retain are exocytosed back out to the plasma membrane surface. So it may absorb things, and things that are not absorbed, debris and so forth, may end up being released. So that lysosome will fuse with a phagosome, food vacuole, if you will, same, analogous terms. We digest the molecules. That basically means breaking down the polymers into simple monomers. And what it will do is it will digest those. We can do this not only with external things like nutrient particles or whole cells like a bacterium. This can also occur in a process called autophagy, where, for instance, a worn out mitochondrion can be done this way. Or if a cell has undergone what is called apoptosis, where it has broken up into smaller vesicles of partially digested cellular contents, cells will come along and phagocytose those and further digest them. It's called autophagy, where you're breaking down your own cells and tissues through this phagocytotic and lysosomal process. And here's a photograph, electron microscope image showing a lysosome. The color is what kind of gives it away here. Again, we can invaginate the plasma membrane if we're undergoing phago or pinocytosis, receptor-mediated endocytosis, and the lysosome will merge with it, release its enzymes, change the pH, and you'll get hydrolysis and breakdown of biomolecules. And here's an example of autophagy, where what's happened is we have an old mitochondria that's worn out. Mitochondria contain lots and lots of enzymes that do not want to be released or should not be released into the cytosol. They can wreak havoc on a cell and trigger cell death processes if unregulated. And so what we'll often see is we'll see a surrounding of that mitochondrion in a vesicle. And then lysosomes will merge, and it will work with another organelle called a peroxisome. We identify peroxisomes because they have these unusual structures inside of them. These are basically protein crystals. They are a crystal of protein. It's really, really interesting. And the pH of the lysosome in combination with the enzymes and the proteins that are crystallized will all contribute to this digestive process in autophagy. And we've been talking about the endomembrane system and phagocytosis and some of those things. Another structure that seems to be involved in some trafficking and or vesicular movement within a cell are known as vacuoles. They seem to be a combination of ER and Golgi. They're larger, more durable vesicles. They seem to be sort of way stations, if you will, in terms of trafficking. They may be very, very important for the structural integrity of a cell, like in the case of plants, or they may be important long-term storage mechanisms for cells that either want to store material that they can't necessarily deal with metabolically, or maybe in some cases some molecules that are only serve a specific type of need. In any case, they're large membranous or of different varying size. Some can be quite large, and then some can be more discrete. There are also vacuoles that serve other types of homeostatic purposes in cells. For instance, in cells that live in very low osmolarity conditions, that is to say, fresh water is low osmolarity. The fresh water has much less dissolved in it than what would be inside the cell. That's going to cause water to want to migrate into that cell by osmotic pressure. And so you have these specialized vacuoles that are designed to keep moving water out of the cell in a way so that what's happening is water's diffusing in. These contractile vacuoles will then pump water out using basically a form of active transport. In plant cells, what we see is central vacuoles. Again, they are sometimes involved in the structural integrity of the cell, help hold the plant up, or they may hold special compounds that are used for chemical defenses in some cases. If you've ever had aloe, aloe vera, aloe is basically mashed up aloe plant cells. And what you do is you bust open all of those vacuoles, and they're full of sort of a semi-oily biochemical that softens the skin and can relieve itching. And then we have some things like phagocytotic vacuoles and so forth. So they serve more than just storage purposes. They may serve structural purposes or homeostatic purposes. So here's a picture of a central vacuole. Maybe it will store nutrients in some cases. Food vacuoles are just part of that whole phagocytosis lysosomal pathway. And here's just a picture of a plant cell with its chloroplasts, and it has that central vacuole. If you've ever sliced an onion, you know how it makes you tear up. Well, that's because when you're slicing it, you're rupturing the central vacuole, which happens to have volatile oil compounds, which can irritate your eyes. Moving on now, we're going to talk about two organelles that are very much involved with energy transfer is the way to best describe it. That is the mitochondria, where we generate ATP and intermediate when it comes to coupling reactions that release energy with the reactions that require energy. And chloroplasts, which are converting energy in the form of light into chemical bonds. We call that photosynthesis. Then thirdly, we have what are called peroxisomes, and I've mentioned those in some of the autophagy that we talked about, or autophagy. These peroxisomes contain enzymes that are often protecting the cells from what we call oxidative damage. When we generate molecules that are reactive, they have the possibility of binding to and altering the shape of things like proteins and other biomolecules, and the peroxisomes are there to help prevent that from happening. In terms of mitochondria and chloroplasts, they have some really interesting similarities. And they also have similarities with bacteria, both in terms of size and in structure. They both have a double membrane of sorts. You have an outer membrane and an inner membrane. They both have their own DNA. It's a circular DNA molecule, and bacteria have a circular DNA molecule. They have their own ribosomes, which are different from the ribosomes we find in the endoplasmic reticulum or the cytosol. They're slightly different, and they're only free. You don't have an ER in either of these cellular structures. And they can change in population, so they can sort of grow and reproduce inside a cell. If a cell needs more mitochondria, the mitochondria can expand or diminish based on the metabolic need of that cell. Pretty interesting stuff. The theory behind the mitochondria and the chloroplast is what we call the endosymbiont therapy. That is to say, there was an ancestral prokaryotic cell that was able to somehow develop sort of an endomembranous structure. It had more infolding of the plasma membrane. That sort of led to organelle-like structures, and that there were other bacteria, other cells that were closely associated. This is a little bit off of this, but if you look at neurons and glial cells, you can almost see a similar relationship. Or if you look at certain types of organisms out in the world, you'll find that one's a fungus and another one is a bacteria or some kind. You do see these sort of these symbiotic relationships, and over time, what may have happened is that these symbiotic organisms were actually ingested and enveloped inside cells. And that with plant cells, you had that happen twice. So you had with animal cells, you had just the mitochondrial precursors being ingested to become mitochondria. But in the case of plant cells, you also had a second endosymbiotic event. And here it is mapped out from figures in your book. You have this sort of, eukaryotic ancestor that may have had a sort of a nuclear-like interior, maybe some infolding of membrane, engulfing some sort of a non-photosynthetic prokaryote, and that becomes a mitochondria. And then eventually the second step, engulfing a photosynthetic prokaryote in the form of a chloroplast. So the basic structure of mitochondria is that it has this sort of double membranous setup. So there's this relatively smooth outer membrane. Again, that's a phospholipid bilayer. And then you have this heavily folded, very, very, very high degree of surface area inner membrane. And then inside this inner membrane is a super dense, semi-gelatinous mixture of proteins, a little bit of water, but it's very, very protein rich, very, very molecule rich, what we call matrix. Not quite as thick as gelatin, but certainly close to it, allowing for very, very regulated reactions to occur. Mitochondria do not do all of the energy transfer, and the cell does not do all of its energy transfer inside the mitochondria. What you have is some aspects going on in the cytosol, and then you move things to this inner space, and then this in-between space serves a very, very specific gradient purpose. It's that in between this space that we get some really, really interesting aspects of energy conversion in the form of potential and kinetic energy. So that large surface area allows for lots of ATP synthesis. Something that's a little bit confounding about mitochondria is they don't always look like these sausages, if you will. They can and sometimes do develop this sort of meshwork-like structure that's a little bit not in the norm. We want to think of it as a nice, hey, that looks like a bacterium that's trapped inside the cell. It's not quite that simple, but nevertheless, the general membranous structure and the fact that you have DNA and ribosomes and that sort of a thing still holds. And that's it for mitochondria right now. We're going to pick up in the next slide segment, but I've left these slides in here so that you can get a nice review in. I really think the detail is good. Make sure you read the book just to pick up as much as you can. And ask any questions that you have about details and that sort of a thing if you're concerned. So here's mitochondria. We'll talk a little bit more about chloroplasts and peroxisomes in the next slide presentation. And thanks for your time. Really appreciate it. Please do let me know what I can do to help you improve learning in this part of the chapter. And I'll talk to you again soon.